to organize a meeting with my best friend and one of the most important person in the world working on peroxynitrite, Rafael Radi. I've been able to visit at his place here and also to meet Francisco. Somehow we met and somehow we started talking about the Tannat. I was not aware of what Tannat was. I never heard about Tannat. I admit, probably because in Italy we have 300 varieties, I was not aware of this one. He described me this one. Actually, we spent a couple of nights also drinking very good Tannat. So I went back to Italy convinced that Tannat was a very good wine and that understanding
We need a lot of bottles. Nos gastamos unas cuantas botellas para convencer.
So anyway, we have been in touch, and last year we started the project that led to the sequencing of the Tannat genome. We got a very nice coverage of the genome. We produced very high quality data, and with this data we have been able to find some very important duplication. I will talk later about the functions of this duplication and the genes that are involved in this area. I just like you to try to, and you Francisco, if possible, to explain in Spanish this point that for me is very much important. So I was comparing before Corvina with Pinot Noir, and I told you that there are 400,000 differences between the two plants. By comparing Tannat with Pinot Noir, the differences are almost 3 million. So I don't know if that is because Tannat came and because the variety that was sequenced is a very old variety, one of the first clones that came here, right, Francisco?
Yes.
But probably because of that, I mean, humans can cross the ocean and mix the DNA, but plants cannot do that. So those plants have been on the Uruguayan soil, separated from Europe for such a long time, that this may be an explanation of this dramatic difference. So heterozygosity is still high, but the differences compared to the Pinot Noir are really astonishing.
O sea que comparativamente con lo que él dice, que tiene casi 3 millones de bases diferentes en los genes, el Tannat con respecto al Pinot Noir. Y ustedes se acuerdan que en la Corvina del Amarone había unos 390.000, o sea que esto tiene mucho más diversidad genómica que la Corvina y que el Pinot Noir. Y él piensa que esto puede deberse a que es una variedad antigua, que puede verse también, nosotros sabemos que es considerada por algunos genetistas el Tannat como una variedad muy cerrada, o sea que no tuvo cruzamientos con muchas variedades. Por eso él da el ejemplo, los humanos podemos cruzar el océano y nos cruzamos con un africano, europeo, lo que sea. Las plantas no pueden cruzarse, entonces el genoma de las plantas es más diverso, digamos, que el que está cruzándose continuamente dentro de la misma especie. Y entonces esto muestra un primer panorama del polimorfismo del ADN del Tannat, que es muy rico, lo cual eso es una buena noticia, ¿no?
Yes, because in Italy they say that a wine to be recognized must be recognizable. So it must be different somehow. And here I have to say the variation, not always to be different means to be better. Okay? So, but for sure in this case the differences genetically encoded differences between Pinot Noir and Tannat are very much high. Anyway, so we then continue the work because sequencing is giving you the structural genomics, so the structure of the genome, but it's not really telling you the function of the genome. In order to understand the function, we have been sequencing also the transcriptome. We started in January this year doing that, and we finished analyzing the data last afternoon. And this analysis is still not ready. There will be more samples coming. But just to let you get the feeling of what is going on here. In yellow, you can see the genes that are used by Pinot Noir. And the second circle represents the genes that we found used by Tannat in the few samples that we analyzed so far. So because of this, the number of genes is so small compared to this one that is bigger, because we analyzed less tissue, less condition. But what is striking to observe is that Tannat is using almost 3,000 genes that Pinot Noir doesn't use.
Conclusión muestra la comparación con el Pinot Noir, que el Tannat tiene genes funcionales, digamos, pasamos de lo que era lo estructural, que era el ADN, a lo funcional, que son casi 3.000 genes más que el Pinot Noir funcionando.
For the Tannat, what we have done for the Corvina, try to assemble the private genome of this variety with the same program, that is the same pipeline, very complicated pipeline that has been used for the Panda, and we ended up with 234 genes that belong to Tannat and that are missing in Pinot Noir. We still don't know in details what those genes are doing, but for sure there is a catalog of genes that is worth a strong investigation in the future.
En la última, esta diapositiva muestra que hay 234 genes que son funcionales en Tannat y que son únicos del Tannat, que lo que falta ahora es ver qué es lo que están haciendo. En este cuadro, muy chiquito, se ve ahí el porcentaje de a lo que está correlacionado a crecimiento, a diferenciación celular, a transporte de membranas, a aromas, a polifenoles, o sea, diferentes áreas metabólicas es lo que indica este cuadro. Lo interesante es que esos genes son 234 genes que son únicos del Tannat con respecto al Pinot Noir y no lo comparamos al Corvina porque no queremos competir con el Amarone.
And now the last 3-4 slides, this for me is very difficult because I'm just a genomicist, so I don't know that much about physiology. So these slides are for you, read them. Anyway, so the thing is, at the end, we found that in Tannat there is a very high level of antioxidant. This plant has an astonishing battery of more than 170 genes which are involved in the flavonoid biosynthesis. There are duplications also in the genome in the area containing genes that are involved in the synthesis of tannins that are giving this so particular taste to the wine, but for me also this is very much important, that are also involved in the antioxidant machinery of the cell. And then there is an impressive set of genes that are involved in